Today we're going to take a little bit of an virology journey. We're going to look at what the general structure of viruses are. We're going to think about how they replicate. And then later on we'll talk about some specific viruses so that you understand how they've evolved, how they work, how they replicate, and therefore how they can cause disease symptoms. So this first session really is just a very simple session, and it's outlining the overall structure of viruses, and it's also going to talk about their general replication strategies. But before we do this, I've got a couple of questions for you. I've called them review, but they're clearly not review. But what do you actually already know? Do you think that the virus, which is structurally the simplest organism, was the first living being, and why? Why do some vaccines such as polio last you a lifetime, whilst there is no effective vaccine against the common cold, for example? And lots of viruses can cause the common cold. One such virus is the coronaviruses, you've got the rhinoviruses. So there are lots of different viruses that cause those common cold symptoms. So press pause, have a little bit of a think about each of those. So the first question, do you think the virus was the first living organism or living being? Well, they're obligate intracellular parasites, so therefore they are required, or they need to be inside a host to live. So therefore it's very unlikely that they appeared before cellular beings in the evolution of life. And when I say lived, they obviously don't live. They are not living as such. Because they require a cell to invade and therefore produce their own offspring, their own new virus progeny. Why do some vaccines such as polio last you a lifetime whilst there is no effective vaccine against the common cold, for example? Well, there are lots and lots of different viruses that cause the common cold. And like I said, the coronaviruses do it and the rhinoviruses do it. And there are over 100 different types of rhinovirus that have been discovered as they strike around the world, and new strains are appearing continually. And it's a moving target that is not effectively combated with a single vaccine because it can mutate very, very rapidly and therefore change, and therefore you don't get that true immunity that you might get with some other viruses where vaccines are very efficient. And if we're thinking about the RNA viruses in particular, and we can think of influenza virus as an example of that, the RNA polymerase that is used to copy that genetic material, that is used to copy that RNA, is very error prone, and hence why RNA viruses are harder to develop vaccines for. And it tends to make an error about 1 in every 10,000 base pairs or so. Now, just to put it into context, the influenza virus is about 13,500 base pairs, so a little bit bigger than those 10,000 base pairs, but it really means that almost every new influenza A virus that is released from an infected cell is different to the original. And that it might just have one mutation, it might have two. Those mutations might make absolutely no difference. They may lead to amino acid substitutions or no changes. But the point is, this is what antigenic shift is. Or sorry, this is antigenic drift. So it's drifting away. So it's mutating and drifting slowly. You also get antigenic shift, and we'll talk about that at a different point when we're thinking about influenza, for example. So here are a few viruses for you to think about. So what I want you to do is again, press pause as always, have a think about what disease name a particular virus may cause and some of the crude signs and symptoms. We don't want very much detail. It's really just to show how much you actually already know. So press pause and have a little bit of a think. Okay, so we've got human papillomavirus. You should know this because you have a vaccine against HPV 16 and 18 in particular. And so that can lead to cervical cancer and to warts, the virus itself. So lots of the skin warts that people have are caused by human papillomavirus because there are over 100 different HPVs. Herpes simplex virus. Of course, you've got HSV1 leading to cold sores and HSV2 leading to genital warts. Varicella zoster virus, chickenpox and shingles. Rotavirus. That's can cause diarrhea and vomiting, primarily affects children. It's an interesting one, the rotavirus, because it's a virus that actually contains 3 capsids, and capsids of a virus are basically a protein core. And so it's got 3 of those to help it resist the acidic environment of the stomach. So it's got those 3 strong structural elements to it to help it live, so to speak. You've then got HAV or hepatitis A, B, and C. Of course, there's D and E as well. And of course, that leads to hepatitis, so liver inflammation. You've got the rhinoviruses, as we've talked about, the common cold and the coronaviruses. So we're thinking about the common cold. So there are common coronaviruses floating around. that are endemic, and they cause common cold. But of course, and they make up about 20% of the common colds that people get. But then you've also got thinking about COVID-19, the more recent disease leading to pneumonia. Then got influenza virus, so obviously that leads to the flu, so that's a lower respiratory tract infection. You've got the Ebola virus, which is a hemorrhagic disease, and it's interesting. Ebola virus, we'll talk about it in a later session, but there's a potential treatment for Ebola virus that they're trialing for COVID-19 as we speak. And HIV, of course, leads to AIDS and leads to immunocompromisation and death due to other viruses and bacteria and infections. So let's think about how we could potentially classify viruses. So we could be thinking about their nucleic acid. Is their nucleic acid DNA, or is it RNA, or is it a mixture? Is that nucleic acid single or double-stranded? And is it circular or is it linear, for example? What about its replication strategy? What about its structure and the symmetry of its capsids? Remember, the capsids are the protein shells that help to encapsulate or protect the nucleic acid material. Does it have an envelope, yes or no? Does it have more than one envelope? The virus I did my PhD on is thought to have 3 envelopes as well as capsid layers associated with it. It's a very large virus. But really, when we're thinking about it, the RNA viruses, as I've said, they generally have high mutation rates, and that's because the RNA polymerase, at least the cellular ones, the host ones, lack the proofreading ability that DNA polymerases have. So if a virus uses the host DNA polymerase to copy its own DNA, then it's very unlikely to mutate rapidly because they've got that proofreading ability. Whereas the host RNA polymerase doesn't have that proofreading ability, so we're getting a change about once every 10,000 base pairs or so. So if we're thinking about how they are actually classified, it was many years ago that Baltimore, Temim, and Dulbecco, they discovered the retroviruses, such as HIV, and reverse transcriptase. And they shared the Nobel Prize for physiology and medicine. And they came across really the classification scheme that is now commonly used for viruses. And it's based on the relationship of the viral genome, whether that's single-stranded RNA, single-stranded DNA, double-stranded DNA, double-stranded RNA, and its relationship into how it makes its messenger RNA. And so if we're looking at these crudely, you can see your double-stranded DNA viruses, and that's like the virus I worked on. So that was a double-stranded DNA virus. Just so you know, its name was African swine fever virus. It's endemic and epidemic in China and other parts of the world, killing off huge numbers of pigs. And as you go through customs in Australia, actually, there are huge warnings, or there were this time last year, about its potential spread into Australia and how it can decimate pig populations. Anyway, that's for another time. So obviously, the double-stranded DNA you can then transcribe that into messenger RNA. You've got the type 2, which are single-stranded DNA. They're then copied into double-stranded DNA, and you make your messenger RNA from there. And as you can see, you can go through your double-stranded RNAs, your single-sense RNA, positive sense. So that means they're reading from 5' to 3', and it's single-stranded and it's RNA. And then you've got your negative sense single-stranded RNA, and your and then you've got your hybrids and your double-stranded DNA, which goes on to single-stranded RNA. So there are lots of potential mechanisms, and we're not too concerned about that. We'll talk about the ones that are most relevant for us. And so really what we're thinking about is the DNA or the RNA, or so rather is the nucleic acid. Is it DNA or is it RNA? Is it single or double-stranded? Is it positive sense or is it negative sense? And what's that method of replication? How do we make that messenger RNA? And that's really how viruses are now classified. So let's take a little bit of a structural overview, and it's really, really crude. And this is for the kind of the simplest viruses, if you like. And so you have the nucleic acid core. That can be, like I said, your DNA, RNA, single-stranded, double-stranded, etc. And that encodes for the transcription of all viral components. So that might be your viral proteins, whether they're structural, whether they're non-structural, whether they're enzymes. So some viruses encode their own RNA polymerase, some encode their own DNA polymerase, some encode virokines that kind of copy chemokines. So there are lots of immune evasion molecules that are also potentially going to be made by our viruses. And so that's shown there. So it's nicely in the center. We've got a nice nucleic acid core. We've then got a nice capsid layer surrounding it. They're often hexagonal, but not always. And that's basically a protective coating made of huge quantities of specific viral proteins. And then we have envelopes. We don't always have envelopes. So sometimes a virus has an envelope, sometimes it doesn't have an envelope. If it does have an envelope, it's derived from somewhere within the cell. That might be the plasma membrane, the endoplasmic reticulum, the Golgi apparatus, for example. So there are, or the nuclear envelope. It can come from lots of places. It basically steals the envelope, the membrane from within the cell and hijacks it and puts lots of its own viral proteins into it. And here they are. Here are some viral glycoproteins that are sticking out. And these viral proteins that are there, they basically can then bind to receptors within our own cells. So if we're thinking about coronavirus or coronavirus, for example, that binds to the angiotensin converting enzyme 2 protein present within our cells. So that's the receptor for the SARS-CoV-2 virus. So the glycoprotein can bind to that receptor, to that ACE2 protein. And then as well as having that attachment to the host, it also, in a lot of viruses, mediates membrane fusion. So it might be the same protein that's kind of changed slightly, or it might be a different protein. And so when those membranes get really close together, that the membrane of the virus can fuse with, for example, the plasma membrane of the cell, releasing this nucleic core into the cytoplasm where it can go on and do its job. Sometimes, just, when we're thinking about this structure, we've got our nucleic acid core surrounded by a capsid. We might have 2 capsids or 3 capsids. Think back to the rotaviruses. But sometimes the matrix between this nuclear capsid and the envelope, and it helps to connect the two. So we call it a matrix. It's sometimes called teguments. There are lots of names for it. So we'll just take a quick look at the overview of the virus life cycle. We know what the structure of the virus is now. So for a virus to get into a cell, it has to bind to an appropriate receptor. Okay, and that's called attachment or adsorption. And so it binds to a susceptible host cell. So the receptor has to be expressed on that cell. We then, at some point, we need entry into the cell. We need to get uncoating so that the genome can enter the host cell and therefore go on to be copied. And that's what the next stage is. We've uncoated our genome, the RNA or the DNA has been released, and we now start to lead to viral gene expression or transcription. And that produces our viral messenger RNAs. Now, viruses might use their own polymerases for that, DNA or RNA polymerase, or they may use the host RNA or DNA polymerase. Remember, if they use the host RNA or DNA polymerase, those are located within the nucleus. So the genome basically in the virus would have to get into the nucleus before it could start to copy itself. Because remember, host RNA polymerase and DNA polymerase are present within the nucleus. So we're making our viral messenger RNAs. We then go on through translation to make our virally encoded proteins. And again, we're hijacking the cell system for that. The viruses do not encode the proteins necessary for ribosomes, so they use the host cell ribosomes to make our virally encoded proteins. So we're synthesizing it. The virus is redirecting cell metabolism so that the viral nucleic acid and proteins are made by the cell, often at the expense of the host cell proteins. So they often switch off and are no longer made. We also have those non-structural proteins, and we get that genomic replication. So that's using our RNA and our DNA polymerases. And then we need to get the structural proteins together to mix with the new genome that's been made. So that's your RNA or your DNA. And then they need to assemble together, they need to mature, and they need to leave the cell to produce these progeny virions. Now, sometimes a cell will explode, and that's called a lytic cycle, and that'll just release everything that's present within the cell. And sometimes they will kind of bud out, so they will use the exocytic cell machinery to leave the cell in a more organized fashion. So if we're taking a look at the virus life cycle in a crude outline, it's characterized by what's called a single-step growth curve. And if we're taking a little look, okay, so we have a look at this figure on the right-hand side. This is the relative virus count. It's done in plaque-forming units, but it just really means the amount of virus. And this is the time. And as you can see, when you get infected by a virus, you have the virus added. And you have this latent period early on. And this is called eclipse. And during this period, you're making the early enzymes. So like I said, things like your RNA or your DNA polymerase. You're starting to copy your nucleic acid and you're starting to make your protein coats. Okay, so those are some of your structural proteins. So in this period, this eclipse period, what you've got, the virus is replicating because you're making early enzymes, you're making nucleic acid, and you're making protein coats. But the cell doesn't look like it's infected, but it actually is. So in other words, we're not making any virus at this stage. It's the latent period, it's the quiet period, but we're getting ready to put that whole virus together. Okay, so the virus is busy with replication. We then go on to the maturation phase, and this is when your nucleic acid and your protein coats and your other structural components all come together, start to assemble, ready to be released. So let's have a little bit of a think about virus attachment and penetration. And we'll use an example. So the example we're going to throw up here is influenza virus. And if we're taking a little look at the influenza virus, this is an RNA virus, just so that you know. It has the hemagglutinin protein and the neuraminidase protein. And we'll talk about those a little bit later on. And so the important thing, I want you to just take a look inside. You can see this is the RNA. 1, 2, 3, 4, 5, 6, 7, 8. So you can see there are 8 separate segments of RNA with the influenza virus. So most viruses have just got one big bit of RNA or DNA. The influenza virus has been split into 8 bits. And this is what allows influenza virus to undergo antigenic shift as well as antigenic drift. Remember, antigenic drift is just random little mutations occurring in little places within the RNA. Antigenic shift occurs when one influenza virus infects a cell and then another influenza virus of a different type also infects that same cell. So it might be a human virus infecting a pig along with a bird virus infecting a pig. And then you can get mixing of those little segments. So you might get this one from the human, this one from the bird, human, bird, bird, bird. And what that means is you've actually then, when the new virus is made, you've got a completely different virus. You've got a big change in the influenza virus that is being released. Now, usually it doesn't make a big difference, okay, or it's not functional. It can't infect humans, for example, but sometimes it can make huge differences, which leads to your pandemics, your potential pandemics, which occur about once every 10 years or so. Anyway, so moving on from that, we'll cover that at another time. So we're thinking about virus attachment. So attachment of a virus to a host cell is highly specific, and it requires the complementary receptors on the surfaces of a susceptible cell and its infecting virus. And the receptors on the host cell carry out normal functions for the cell. So they might be involved in the uptake of proteins, for example, through receptor-mediated endocytosis. And receptors include proteins and carbohydrates and glycoproteins and lipids and lipoproteins, or various complexes of them. What does influenza use? Well, influenza uses its hemagglutinin, and its hemagglutinin grabs on to sialic acid, which is like a small sugar that is attached to many different proteins on the cell surface. And it does this obviously in your respiratory system, in your respiratory tract. So the HA grabs hold of sialic acids on the cells within your lower respiratory tract and gets into cells that way. Neuraminidase, which is the other one, that's there. Actually, we'll cover that when we deal with influenza virus. That's to help the virus get out of the cell. So it's for the new virus to leave the cell, okay, when it's been made. So attachment of a virus to its host cell results in changes to both the virus and the cell surface that facilitates penetration. So in other words, you can get the two, if it's an enveloped virus, you can get what's called receptor-mediated fusion. So again, here's we've got a nice little virus with an envelope, and here's our envelope and our envelope proteins, and here is the receptor that the virus binds to. And as the virus particle gets closer and closer to the plasma membrane, in this example, of the susceptible cell, that membrane fuses with this one, as you can see there, the fusion of the viral envelope with the cellular envelope, releasing the nuclear capsid inside the cell, and the membrane, the envelope of the virus, is left on the plasma membrane in this case, and here are all the viral proteins left on the plasma membrane. But it's obvious from this that only viruses with an envelope, such as the coronaviruses, can enter through receptor-mediated envelope fusion. And just so you know, a permissive cell, it's just a definition, is a host cell that allows the complete replication of a cycle of a virus to occur. So we also have a process called receptor-mediated endocytosis. And this is where a virus is taken into the cell, into an endosomal compartment, a bit like a lysosome, but it's called the endosome. And uncoating often occurs in that acidic environment of the endosome. Structural proteins might become rearranged, for example, and the nucleocapsid then injected into the cytoplasm. And so in this case, what you might have, it can be enveloped as well as unenveloped. You don't always get envelope fusion with envelope viruses. So again, the virus binds to the cell, it gets taken into this endocytic vesicle, then into an endosomal-like compartment, which has got lots of hydrogen ions because it's nice and acidic. That changes structural conformations, and sometimes there's a little weak point in a virus. So the virus I worked on had a little weak part to it where the acidic environment, or the redox environment rather, started to influence its structure, and that allows the DNA or RNA to be released into the cell interior. So viruses, obviously, that can't enter cells by a receptor-mediated membrane fusion usually enter via this mechanism through receptor-mediated endocytosis, where the virus is taken into the vesicles and delivered to the acidic environment of the endosomal lysosomal compartment. And it's interesting, like I said, we'll talk about influenza another time, but it is interesting that influenza has an envelope but still enters the cell through a mechanism similar to receptor-mediated endocytosis, not receptor-mediated membrane fusion. So what about the proteins that are present? Because during a virus life cycle, it makes some early proteins and it makes some late proteins. So the early proteins are made very soon after the synthesis of viral messenger RNA. So they're synthesized very soon after infection. They're often necessary for the replication of the viral nucleic acid. So they might be your RNA or your DNA polymerases if the virus makes its own RNA and DNA polymerases. So they're typically acting catalytically as enzymes, and they're usually made in relatively small amounts. But it's also the time when they start to make immune evasion molecules. So molecules that are starting to fool or switch off the immune system so that you don't stop that infection. Okay, so you don't stop the virus from replicating. And there are lots of mechanisms for that. And in particular with the large DNA viruses, they tend to have a lot more immune evasion strategies because they've got bigger amounts of nucleic acid and bigger information and more genes. So, they might express 100 or 200 proteins. So a lot of those might be involved in immune evasion strategies. Whereas other viruses may only express 5 or 6 proteins, which is basically the structure of the virus. So they don't have those immune evasion strategies. And then you've got the viral late proteins. They tend to include the proteins that the virus coats. So that'll be some of your membrane glycoproteins or your capsid proteins. They tend to be structural in nature, and they're synthesized in huge amounts, and they completely overwhelm the host protein machinery. So they make vast amounts of the protein, and it can all start to assemble together. So I mentioned some of these immune evasion strategies a couple of seconds ago, and there are lots and lots of mechanisms that viruses use to help evade the immune system. And a couple of examples I'll give you, things like what are called virokines. And so virokines, they basically mimic cytokines or similar extracellular immune regulators. And so if we've got a virokine, that can come along and bind to its receptor or bind to a cytokine receptor and therefore block the action. And if you block the action of the cytokine, it can therefore no longer go on and do what it would do, which helps to stimulate the immune system. There are also viroreceptors. And so viruses may produce viral proteins that are released into the blood, for example, or into the tissue that basically bind to cytokines and prevent them from having their action. They've taken the cytokines out of use, out of action. So this is your example here. You have a chemokine, and that then goes on to bind to a viroreceptor, and it can no longer bind to its normal receptor. So therefore, you're not getting that signaling pathway being activated. Now, there are dozens and dozens of different immune evasion strategies employed by viruses, like I said, particularly by those larger ones. Other immune evasion strategies may include going dormant, for example, the herpes viruses. And like I said, some others attempt to modulate any immune response using virokines and viroreceptors, for example. And like I said, most of these have been found in the large DNA viruses, such as the pox viruses, you're thinking chickenpox, or the herpes viruses. So what about virus assembly? So virus assembly is quite a complex process because the viruses do form very structural complexes. And a lot of the time they seem to self-assemble. So the viral components seem to spontaneously organize themselves, for example, in helical viruses. Whereas other viruses can have more genetically or viral controlled assembly. So the assembly may be directed by scaffolding proteins, helping to bring proteins next to each other, chaperone proteins, which are involved in folding specific folding. So we have what are called heat shock proteins in our host cells, and those are involved in helping to fold all our proteins properly. And so I'll tell you about my little virus again. So I worked, like I said, on African swine fever virus, and I discovered the first virally encoded chaperone protein. So it helped to fold the main structural proteins of the virus I was working on. So they may be chaperones, they may help to scaffold complexes together, and then, of course, there may be enzymes as well involved in polyprotein processing. And the genome becomes packaged into the capsid. So then we have naked viruses. So we're thinking about virus assembly and release. So pre-assembled capsomeres, that's just small subunits or small protein complexes joined to form big, large, empty capsids, which are the precursors of the virions. And then the nucleic acid then can be incorporated into that empty capsid. And with envelope viruses, the viral proteins are first associated with the nucleic acid to form a nucleic capsid, and then that becomes surrounded by an envelope. And as I said before, that viral envelope has the lipid composition of the membrane where its assembly takes place, whether that's the nucleus, plasma membrane, Golgi, ER, or other intermediate compartments in between. And so the whole point is viruses, they make proteins, they hijack the host cell machinery to make proteins, but they don't make lipids. Okay, so they don't stimulate lipid synthesis, but they do hijack and steal membranes from within the cell itself, if they're envelope viruses, of course. So if we're thinking about a crude overview of replication, a virus enters the cell by binding to a specific receptor. It comes into the cell, and then it may get in through receptor-mediated endocytosis or receptor-mediated fusion. But the point is it releases its genetic material into the cytoplasm or into the nucleus. So it then starts copying. It starts making your early proteins. So you've got your messenger RNA and your early proteins. And then it'll copy the RNA or DNA, and then it'll make the late proteins. And then all of these things will come together. The new nucleic acid and also the structural proteins will all come together to form the mature virus particle. Now, the example you can see here is actually influenza virus, and you can see that because you've got the different RNA segments there. You can also see that you've got the HA and neuraminidase there, so you know that this is actually looking at influenza virus. But the general structure, the general replication strategy is the same. So just before we finish off, we'll just talk about the basic patterns of infection. So we have apparent infections, localized infections, and disseminated or systemic patterns. So in a localized primary replication, the virus replication remains localized near the site of entry, for example, within the skin or within the respiratory tract. And you tend to get just localized damage in those cases. But of course, sometimes the virus can spread, and the virus may spread by cell-to-cell contact through the bloodstream, through the CNS, or through the lymph. It tends to have a longer incubation period, and it can be associated with severe disease in target organs. So really all I'm wanting you to think about here is using a couple of examples that you will know about and will have heard about. Do these viruses, and we're thinking HSV1 and HSV2 and HPV, do these viruses cause localized or potentially systemic infections and symptoms? So all I want you to say in each case is can it be only localized or can it be systemic as well? And if it can only be local, obviously you've only got a primary replication site. But if it can go systemic, so if it can go elsewhere, what is a potential secondary replication site? So what I'd like you to do, just have a little bit of a press pause, have a think about each one, and then we'll go through it. Okay, so let's go through HSV1. Are you thinking that's potentially localized, or could it become systemic? Well, the answer is it could become systemic. What's that primary replication site? Well, as you'll know, it tends to be the mouth and around the throat. Secondary replication site, so hopefully you already know this. It can be, secondary replication site is within the central nervous system. Interestingly, herpes simplex virus 1, so you're thinking about your cold sores, obviously, is in about 30% of healthy children, and about 60% of healthy adults have herpes simplex virus, either active or dormant. So what about herpes simplex virus 2? So that's where you're getting sores down below. So again, can become systemic. This time we're thinking about the urogenital tract, and again, secondary replication cycle location is the CNS. And HSV2 is thought to be in about 3% of healthy children, so quite a large number actually, and about 25% of healthy adults. Lots of people don't have symptoms of HSV2, but yet will be actively shedding the virus. So during intercourse, they can pass it on. Even if they don't see anything there, they could be actively shedding HSV2 and passing it on. So something to be scared about, clearly. Anyway, so what about HPV, human papillomavirus? Well, that is localized. And where's it localized to? Well, the epidermis and the skin. So just one extra little thing on HSV1 infections, because there's some really interesting stuff that's coming out more recently. There's accumulating evidence now suggesting that HSV1 infection of the brain, both in symptomatic and in asymptomatic individuals, can actually lead to neuronal damage and eventually influence neurodegenerative function and lead to neurodegenerative disorders. So it's quite interesting how time and our knowledge base is moving on all of the time. So really just a very simple question for you at the end. It's been me just blurbing on up to now. So a couple of questions, press pause, have a little bit of a think. They're nice and easy. I would hope you get them right. And I'll go through them. Okay, so enveloped viral membranes are generally blank with associated virus-specific blank. I'll give you a clue. Only one of those is correct. Which is it? So enveloped viral membrane proteins are generally lipid bilayers. So that means it's either number 1 or 3 with associated virus-specific proteins. So the answer is 3. So it has lipid bilayers that it's stolen from the cell and its own viral-specific proteins that have accumulated within a specific membrane compartment. Cellular receptors for viruses may be composed of which of the following? So basically I'm saying which ones are correct and which ones isn't. So more than one, I'll give you a clue, is correct in this case. Okay, so can cellular receptors for viruses be proteins? Yes, they can. Coronavirus and ACE2. Carbohydrate? Yes, they can. We're thinking of sialic acid in influenza. Lipids? I'd give you an example, but I can't think of one. But the answer is yes anyway. Combinations of proteins and carbohydrates? Yes, clearly. And combinations of lipids and proteins? Yes, clearly as well. So basically, pretty much anything that's being expressed on the cell surface that is specific to that cell type can potentially be used as a receptor for a virus to get into the cell.